right, and our webinar is now recording. So everyone, welcome. For those of you who are just joining, we'll have a few logistics things here at the beginning, so you're not going to miss a whole lot. Let's get started with our webinar today, discussing eMass, a computer-assisted tumor response assessment tool for oncological clinical trials. And a little later in the webinar, we'll have some chats about how that could get even more interesting with some other things. So from a logistics perspective, we've got about 45 minutes of presentation. You're actually going to see a live demo of the eMass solution, which is always a little bit thrilling when you do live software on a live webinar. So yeah, that's always a little exciting. There'll be times for questions and answers at the end if we're able to do things in a timely manner. What I would tell you all is this. On your GoToWebinar panel on the right-hand side, you will see a question box. You are welcome to type in questions to us at any time that they dawn on you. You don't have to save them till the end. What we will do is save them till the end and try to answer as many as we can at the end. So feel free to type in questions anytime. And because I did remember to press the record button, woohoo, good for me, we will have a recording of this available at Imaging Analytics University. So without much further ado, let's introduce our two handsome gentlemen who here will be giving you most of their content. First, joining us from the University of Mississippi Medical Center, Associate Professor in Radiology, is Dr. Andrew Smith. Andrew, thanks very much for being here with us. Really appreciate it.
Glad to be here.
Also with us is Amit Vasanji, the Chief Technology and Science Officer here at ImageIQ. So by way of background, just so you all know who's going to be sharing their insight with you, Dr. Smith is a practicing radiologist at University of Mississippi Medical Center. His research focuses on the development and testing of new imaging biomarkers for the detection and staging of cancer, chronic liver disease, stroke, and osteoporosis. Spent a lot of time on the educational circuit here in the lovely state of Ohio, receiving his MD and PhD from the Medical College of Ohio in Toledo, about 2 hours west of where Amit and I are sitting, and did his fellowship in abdominal imaging at the wonderful Cleveland Clinic. Numerous publications, basically 50 or more national and local media conferences, multiple research awards, and it's wonderful to have him here. We work with Andrew in a number of ways at ImageIQ, and that's absolutely great. He also serves as the president of Radioptics, a group that does reads for the clinical trial market, which is really great, and I know we work together in a lot of ways that way too. Amit is here. He is basically our Chief Technology and Science Officer and founder of ImageIQ, with over 15 years of experience in basic and clinical research around image acquisition, processing, analysis, visualization, software programming. He was the founder of the Cleveland Clinic's Biomedical Imaging and Analysis Center, although you and Andrew never really intersected at the Cleveland Clinic, which is kind of interesting. It's a big place. He's authored many publications, over 50, in some high-impact journals, including being primary author on funded grants, both at the state, local, corporate, institutional level. It's great. He graduated from the University of Miami, not Ohio, but Florida, which is wonderful, and got his PhD from Case Western Reserve University. I'm Tim Colbago, who's talking to you. I'll be playing the role of moderator here today, and I'm the CEO of ImageIQ. Very quickly, just about ImageIQ, very, very shortly. ImageIQ is kind of this wonderfully interesting blend of an imaging CRO with a software company where we basically love to help people with their imaging problems and help them extract quantitative data from their imaging, move images around for their clinical trials and their preclinical research. And that's what we do. We're located here in Cleveland, Ohio. We were lucky enough to spin out of Cleveland Clinic, and we're sponsoring the webinar today. That's why it's free for everyone. We get a lot of those questions. The webinar is free? We're like, yeah, it's free. Why not? We gotta love to share the knowledge. So that's the deal. As we get a number of people who are just coming in, as their 10 o'clock meetings Eastern time kind of ended and they're rolling into this, we'll start with a little bit of a story. If you've ever attended an ImageIQ webinar before, we like to start with stories. And this story is about a friend of mine who I used to work with, and we had some business in Europe. And he was heading off to Europe. And he said, hey, listen, when I get there, to give you an idea of the vintage, he had a BlackBerry. And he said, hey, when I get there, I got the international plan on my BlackBerry. I'll give you a call and let you know how the meeting went and all this kind of stuff. I'll call you after the meeting. I'm like, this is great. I'm like, just let me know. So I'm waiting for the call. The call never comes, right? Finally, I get an email from him, like, you're not going to believe this. I'm like, what happened? He goes, well, I was using my BlackBerry on the plane. You know, he was playing games or whatever he was doing. He goes, I got here and I went to charge the BlackBerry, and well, and I mean, as he was telling me, I'm like, oh Lord, you plugged it into a European outlet without checking if the transformer actually handled 240 volts or whatever, right? He's like, yeah, I fried the whole. Oh, he fried his BlackBerry. And we thought, you know, that's kind of funny. If you look at this slide here, you know, you see that a lot of the adapters around the world are kind of interesting for power. I mean, we all need electrical power. Everybody uses it, but the adapters are interesting. And just because you can get something to physically fit into an adapter doesn't mean it's always going to be a good idea, right? And it might fry your BlackBerry. And so, you know, we really thought as we were talking about eMass and putting this webinar together, you know, we were talking about all the different ways that you could do tumor assessment and figure out, well, what fits, right? And we thought that was kind of a fun, so it was a funny story, and I like bringing it up because I like giving the guy who fried his BlackBerry a hard time. But our learning objectives today are really interesting, right? We're going to talk a little bit about the history of tumor response assessment. We're going to talk about those challenges in the current methodology. For those of you who are on the webinar, you've probably heard of RECIST 1.1 or the Choi criteria or something like that. You'll hear about that, what those challenges are, and then talk a little bit about eMass and get into that along with the demo so you can see how it works and what value it can add. And then we're going to have a little bit of a conversation about the potential of where this can go, because it's certainly in different, you know, software is never done and the vision is pretty broad. So we're going to be doing that kind of stuff. So with that, I will turn it over to Andrew, who can walk us through a little bit of history on tumor response assessment.
Sure, appreciate the introduction. Thanks so much, Tim. So we're going to talk about tumor response assessment history here, and we're kind of taking it back to the beginning. Here we go. There we go. So in the beginning, when we started with tumor response assessment, we're talking about the World Health Organization, or WHO, published tumor response criteria, and that came back in 1981. You'll recall that was bidimensional axial tumor measurements. And then from there, we're looking at RECIST was what popped up next in the year 2000, and that was unidimensional axial tumor measurements, up to 10 target lesions, a little bit better definitions, and that was very much the standard of care for quite some time. And then RECIST 1.1, which is a bit newer, came out, and that was just 5 target lesions from there. Now, these are like the core criteria that we use for tumor response assessment, but a lot of other criteria have popped up, and so we're going to talk about some of those now. So, for example, the European Organization for Research and Treatment of Cancer had PET recommendations that came out in 1999. And then Choi criteria that Tim mentioned came out for metastatic GIST on targeted therapy in 2007. And that took into account not just tumor size changes, but also attenuation changes. Some people say the word density, but more correctly stated, it's attenuation. A number of other criteria have followed this, modified Choi criteria, something called modified response criteria, the immune-related response criteria, mass criteria that I helped develop, 10% tumor diameter criteria, et cetera, et cetera. And then even more came out in more recent years. Now there's like CT texture analysis, which is an advanced way of quantifying tumor intensity and intensity changes. And then we more recently developed vascular tumor burden, which is coming to a theater near you when we're trying to publish that in radiology. It was just accepted recently, and we'll kind of cover that as we go forward here. Now, each of these criteria have a number of different problems. I can't go through all of them, but you know, with WHO, there's target lesion selection's a little bit unclear. How many you're supposed to select? Really, you select them all. There wasn't really a great definition for minimum size threshold. The definition of progression was a bit unclear. It's very difficult to perform with WHO just because of the number of target lesions. RECIST is similar. It has 10 lesions, which is a bit too many. You're going to measure, you know, the long axis of most lesions, actually for all lesions in RECIST 1.0, including lymph nodes. And the reality is, in real clinical practice, we tend to follow the short axis of lymph nodes. And so that's what got revised when we got to RECIST 1.1, as the lymph nodes were revised. We now measure those in short axis, but there's still problems with RECIST 1.1. How do you handle necrotic and cavitary lesions and cystic metastases and things? So there are problems with these criteria, and they get very complex. So one thing we want to do is just kind of quickly review RECIST 1.1 with you. It forms a common language for objectively defining tumor response, and it's the most widely accepted criteria for assessment of metastatic disease. And the process typically follows that you obtain a CT or MRI, you identify your measurable lesions that are greater than 1 centimeter on the baseline exam. That's for the target lesions. You select up to 5 target lesions with up to 2 in any one organ. and you're going to want to measure the long axis of the target lesions to calculate the baseline sum, except as we said for lymph nodes where you measure the short axis. And it's kind of a little quirk that you have to know that the lymph node has to measure at least 1.5 centimeter in short axis on the baseline. And that's kind of different than clinical practice. As a radiologist, I can tell you we tend to focus on lymph nodes that are 1 centimeter and larger. So RECIST has strict guidelines, and you need to follow those closely. Then you need to assess your overall response of the non-target lesions, and then categorize the overall disease response. And so you're probably very familiar with this, that RECIST has complete response, partial response, stable disease, and progressive disease. And it has very strict definitions for those. You know, complete response, disappearance of all lesions, partial response, tumor shrinks more than 30%, et cetera. And so that's kind of how we do it, and it's a little more complicated than that. I've really simplified it, sort of made it sound really easy, but the reality is it's not easy. You have non-target lesions, you have lesions that sometimes split or coalesce, and you have these cystic lesions. A lot of funny things can happen. You can have missing data points, and how you handle those gets very complex in a clinical trial, especially as you go forward in time in a longitudinal analysis. You can also calculate your best overall response, and you can also get the duration of overall response from that. So we'll give you an example of here what we're talking about, and then you'll see why this comes into play when we talk about eMass in the future. So here's just a standard patient, 5 target lesions. Maybe they have other potential metastases, but these are the ones that were chosen as target lesions. You can see most of these are metastases, and they're lined up in their long axis, except for the lymph node, which is turned sideways in a short axis, because that's what you would measure in this particular patient. So their total sum is 10.5 centimeters. Let's say they go through chemotherapy and we're out 3 months, and now the tumor's shrunk a little bit. It measures the total burden is now 8 centimeters. Well, that's a 20% decrease, so that would still be stable disease. They haven't hit enough change to call partial response at this point. If they continue to shrink, you're still going to compare to the baseline. So this is now a 40% decrease compared to the baseline, and now it's partial response. And so that's good. Now they've hit partial response. Perhaps after that, ideally they would stay at that or continue to shrink, but this particular patient is not, and the tumor starting to grow again. So now we're going to compare to the lowest tumor burden, and now it's a 17% increase. Not enough to call progressive disease, but we're headed in the wrong direction here, and perhaps that continues on. And you'll notice that here, this is a 33% increase since the smallest tumor diameters, and that's enough to call progressive disease. It does not have to get all the way back to the original size or be 20% higher than that, just 20% greater in size than the smallest tumor burden so far. And so from this, we know our best overall response in this case was partial response, and you can calculate your duration of overall response. So that's kind of an overview of RECIST. I've really simplified it, as I said. It's much harder than that in real clinical practice. And now imagine in the world that we're in where a lot of times you're using new drugs and other criteria become important, whether that's an immune-related criteria or if you're using an anti-angiogenic, there's a number of different criteria you might use, or a number of these targeted agents. And so here's just a list of some of them. These are the ones that will eventually I'll show you that are currently in eMass, although we can add anything to that. And you'll notice we have VTB criteria, vascular tumor burden criteria, RECIST, 10% tumor diameter shrinkage, CHOI criteria, et cetera, listed on here. And each of them have different definitions. They each have different thresholds. So I can tell you, when you have a clinical trial and you have to analyze multiple of these, and we have one right now between ImageIQ and Radioptics that we're working on that does this, it gets very difficult to keep track of all these things because it's just very overwhelming to make all of the measurements and then make sure you categorize the disease correctly when you have multiple criteria. So I'm just telling you that's a major challenge in these trials, and we're trying to show you a solution for that we've developed. So I'll let Amit kind of comment here on some of the challenges and confusion and regulatory compliance that we see in these clinical trials.
Great. Thanks, Andrew. As Andrew showed, there's a lot of different criteria that you could use in these trials. So if you're a centralized reader and you're reading for multiple clinical trials and they all have different criteria that they're using, you could get pretty confused on which ones you're supposed to use for the trial or the case that you're working on. And it's particularly confusing if you trial all of those criteria that Andrew mentioned. They're not necessarily required by the FDA. You can choose your own metrics for your trial as long as you can justify it for your pathology or for your endpoint. So that can add to the confusion, right?
Well, and it also is, a lot of folks who do the reading for clinical trials, right, are reading for, it's rare that you will find a radiology team that will read for one clinical trial, right?
Correct.
So on this trial, they're doing RECIST criteria. On this trial, they're doing some other criteria. Now they're getting confused between, well, which one was which for which, and now you're ending up in all sorts of weird, right? I mean, that doesn't help. And the other challenge is clearly the regulatory stuff, right?
Right, and you probably, if you run some of these trials, you may not even know you have these compliance issues. So in general, a radiologist will use whatever they're comfortable with in terms of making these measurements for the different criteria. So they probably are going to use something that's attached to their PAC system, or they're downloading some software like Osiris and they're making their measurements. But as they make these measurements, they're not being audited, so they'll draw along the long axis of a tumor, but nothing's being really saved, right? You could actually force the save. You can save an overlay or an annotated image, but that's not really tied back to the original case. So, and let's say a different time point comes along, you have something 6 months later, and you're making the same measurements for these target lesions. You have no way to figure out which ones you made the measurements for, and if you had to go back and make a change to one of them, it's really hard to bring that back up and make those changes.
Yeah, absolutely, and it's also kind of funny. Probably our biggest webinar we've ever done was compliance in imaging. It was a couple 3 years back. I mean, it was a while back, but just the challenge around being compliant in imaging and you got to manage the audit trail. You got and the systems. Andrew, we talked about this even on the rehearsal when we rehearsed this a little bit, right? The systems that you use for diagnostic clinical care radiology are very different are designed for a different workflow than what you are doing in clinical trials and that kind of research. So they have different features, functionalities. You're going to see some of that in eMass.
Right. Yeah, you really need something that's trained for the actual application. And one of the issues that we'll talk about later is as you make these measurements, they're not necessarily being put directly into a CRS. So you're writing these down, you're putting them into another system, another EDC, another electronic CRF, and that causes transcription errors and variability that could lead to adjudication.
You got it.
All right, yeah, you kind of led into it there nicely. That as radiologists and even as the oncologists view these, we're used to looking at like PACS workstations, and those are designed to handle any type of image in any situation. They were not designed for clinical trials, specifically for metastatic disease, for archiving, making a visual and a data archive of images and keeping up with the regulatory compliance. And so that was the need that we saw, and that's why we developed what we call computer-assisted tumor response assessment. And that's really what eMass is about. It's really going to help the user provide guidance and set rules so that we can prevent errors in these clinical trials. So here are some of the errors that eMass prevents. And these may seem very basic, but when you're looking at a large clinical trial and you have multiple readers potentially, or even one reader doing this over time, it's very easy to make these errors, which one would be selection of more than 5 target lesions, or selection of more than 2 targets in an organ, selection of a metastasis that's too small, or a very easy one is a selection of lymph nodes that are too small. That's very common because, as I said, radiologists tend to choose lymph nodes that are over 1 centimeter in short axis, and that's not the rules for RECIST. There's also like data transfer errors. Now that when we're dictating in real life as a radiologist, I'll say 3.1 centimeters, and then I look over my screen and it says 2.1. That also happens when in clinical trials, you make measurements, you see something on your screen and you need to write it down, and errors come, whether it's a decimal point error or just a number error. In addition to that, you can have mathematical errors, which is to say when you're adding up the sum. You would think that that's very simple, but I can tell you, as you have multiple time points, things add up and mathematical errors occur, and that can really change what the outcome of a trial is going to be if you're having those kind of errors. And it's also a source of variability where perhaps one person's a little more studious at making sure to keep track of their math and others are not. So when you have multiple readers, it becomes a problem. The last one's really response classification errors. You can actually have all of the math correct, get to the very final step, and then just choose the wrong category. Now that may seem simple, and how could that happen? But imagine now that you have a clinical trial where you want more than just RECIST, maybe just one other criteria, or 2 or 3 is some of the ones that we do. It gets very confusing then when you have to choose the right classification. And so that's what we're preventing with eMass. It's software that prevents those errors. And it also allows for some improved workflow. So it does simultaneous measurement of multiple tumor metrics. We'll show you how it does that here in a moment. It does simultaneous assessment by multiple tumor response criteria. So you're really evaluating by multiple criteria simultaneously. it actually reduces reader time. So you're saying, wait, you can reduce all these errors and do all this stuff in less time. That's the point. This is not PACS. It was specifically designed for metastatic disease response assessment. And yes, we can archive the data, the images, the regions of interest. We can perform advanced analysis of the regions of interest, like CT texture analysis. And we can generate an instant summary report. So we'll kind of show you a little bit about what that looks like here. So most of these criteria are based off of RECIST 1.1 selection of target lesions. So that's a good thing, because they all tend to have up to 5 target lesions. And then you have to categorize the software. In the software, you'll categorize, is it a metastasis or is it a lymph node? And that becomes very important because they're handled differently in terms of their lengths. And what we do is, what you're seeing here in the color is, that's the region of interest we're drawing around these tumors. So the tumor on the left is like the baseline, the tumor on the right is the same tumor, but after a round of anti-angiogenic therapy in this particular one. And so you're drawing a region of interest around this, and it's automatically deriving the longest dimension. Also the short axis if it's a lymph node. It also keeps track of the area, and what's shaded in red here is the vascular tumor burden. That's something proprietary that we have created that's unique to us, very useful for anti-angiogenic therapies, really all targeted agents, including the immune response agents. We think this is going to be very useful for that. And what I'm not showing you is you can calculate thousands of different CT texture analysis parameters off of this as well, which can be very useful in a research setting. So that's quite a bit of different things that we can do. At the conclusion, a summary report's instantly generated. So this is what the summary report would look like here. And basically, it tracks all target lesion data, images, and regions of interest. All that is exported in the background. This is just the summary. This is not everything, but it's a nice visual summary. And what you're seeing here is the patient information in the top left. Of course, we blinded this. This is a real patient from a real clinical trial. This is not Photoshop. This is actually what pops up. I'll show you that when we do our example here. It has vertical display of the target lesions. Now, you could do that graphically if you wanted to, especially if you're doing this over time, over multiple time points. But you can actually see the actual tumors here, see their size in relation to one another, and that, the lymph node's turned on its side. You notice target 3 is a lymph node. It's turned on its side because it's the short axis that you'd be tracking. And so it gives you a visual representation of it. I think that's very useful to the oncologists. Our oncologists are giving us feedback on this and liking that. So it also tracks all tumor metric data. If you look at the bottom, you can see all these metrics on there, the hardcore metrics. But maybe you're a summary guy and you like the big picture, so look in the top right, and you have your percent change in length and mean attenuation, area, vascular tumor burden, et cetera. What happened to the non-target lesions? What's going on? Are there new metastases, yes or no? And then automatically, this is all done automatically, because as you go through the software, you answer the critical components necessary to answer what is the response here. And so we can see those responses here according to each of the different criteria. And of course, it could be different. One could be partial response, another could be stable disease, and so forth, depending on the criteria. And so eMass does all of that for you. And then what's really cool about eMass is you can integrate this into an electronic data capture device that ImageIQ has. And I'm going to let them tell you a little bit about that and how that could help you in a clinical trial.
Great. So you want to?
Oh, yeah.
Sorry.
Yeah, right.
So if you run a clinical trial, you probably used an electronic data capture system or a EDC. It's essentially, in the form that you probably use it, is an electronic CRF. So you have a study coordinator log in, or you have a reader log in, and they enter the information about the patient and the time point. What ImageIQ has developed is called EDC-IQ, but it's an imaging-centric EDC. So it basically handles the workflow for imaging throughout the trial for every patient, every time point. So just to give you an example of how this works, you would have a study for a given trial, for a study coordinator for a given trial upload images into the EDC that gets stored on the cloud. That queues or triggers a QA, a person to actually log in. They get notified, they log in and do their QA, all within the EDC. So they don't have to download the images, they don't have to download anything special. They log in with their unique username and password, and they can QA and fill out a CRF. That then triggers a reader. You could have 3 or 4. It triggers them all simultaneously to go in and log in and look at their work list and start assessing a particular case and time point.
Right.
They can then make their measurements directly in the EDC. So everything that you'll see Andrew presenting in his demo for eMass is basically going to be in the browser. So they make all of the measurements for the tumors within the browser. And what's unique about that and what's special about it is that as they make their measurements, each of those metrics, and you'll see a bunch of them pop up as Andrew makes his circles around his tumors, those all get populated into the database automatically. So no one's going in and copying and transferring and transcribing data to another form. It gets populated automatically as they do that, and it error checks as it goes along. So you probably run a clinical trial where you have to do a bunch of queries at the end of the trial. This prevents that because it's doing the checking live as the reader is making their measurements. As that's all being done in the system, they sign off on it. It's saving all of those regions that they're drawing. So, for example, if this case had to get adjudicated, the adjudicator can log in and he can see both regions of interest, and he can decide whether he wants to choose one or the other or make a new one. If the reader decides to log back in and make changes to their regions of interest, they can go ahead and do that, and it audits that entire process.
Exactly. So the idea behind what you're going to see here, and I'll press the next slide here, so we'll get Andrew set up to do the demo, because hey, why not on a live webinar do some live software demo? That'll be super cool. Just to let you all know, we're going to go over the demo so you can see how this works and see what happens, and that's really very cool. Hold on, I got to press the button here. Don't go anywhere. There we go. But you may be asking yourself the question of, okay, well, how do I, what's the availability of eMass? Where is it in the process? What you're going to see here is the workstation version of eMass that Andrew is using in his research. If you're interested, you're a radiologist, and you're interested in this kind of research and figuring out how to be part of this, you're welcome to contact ImageIQ or Andrew to get that conversation done. If you're running a clinical trial and this concept of eMass and quantitative data and having all of this stuff available for you is interesting, you're welcome to give ImageIQ a call. We've got a couple of incarnations of it that we can show you. So with that, we'll turn over the demo to Andrew and let him dazzle us with the eMass amazingness.
All right, I like it. Sounds good. Well, bear with me, everyone. We're going to actually show the nitty-gritty and the details of this because it is fairly fast, and we have time to do that. So this is the overall display here. We're going to go ahead and load up a case. Okay, so CT1 is our baseline case. So I'm going to come in here and find a patient, the de-identified information here. In this particular patient, they had separate acquisitions of the chest, abdomen, pelvis. Sometimes that's how things are done. So I'm going to load up the chest. I'm going to find the abdomen here, and I'm going to go ahead and load that up as well. And also this person had a pelvis here, it's done as well. So we're going to go ahead, and that's going to stack them up for us. It makes my job as a radiologist easier. I can scroll through here and confirm that, yes, these do have metastases that I'm seeing, and also there's intravenous contrast. That becomes important because some of the criteria, you need to know that information to be able to calculate, like, for example, Choi criteria. So we're going to say, yes, that does have contrast enhancement. I'm going to go ahead and load up the follow-up exam here, which is cycle 1 for this patient. And we're going to go ahead and load the chest, abdomen, and pelvis after one round of therapy here. So I've got those loaded up here. Again, it asks me, is this contrast enhanced? And I can look at this and confirm that, yep, definitely contrast enhanced. So I'm going to leave that defaulted to yes. And we got our case open here. We'll just take a quick look through here and kind of see that there are some metastases. Here's a big lymph node one. Here's some lung metastases. We can change the windows as we need to take a look at those. And come on down. We're still in the chest acquisition, but we're going to get into the abdomen here in a bit. Here's a big retroperitoneal mass that we'll come back to and some other ones. Let's go up and start making some measurements and things. Okay, let's go ahead and pick a target lesion. I'm going to pick this one right here. Okay, so we'll find that over here.
And Andrew, as you're doing this, at this point, right, nothing you've seen here is any different than any traditional PAC software you would use, right? Where the eMass value is kind of right. This is where the rubber hits the road relative to the eMass value, right?
Yes, it feels like a PACS. We've designed it that way. It's very easy to change windowing and so forth. So what we have here, things that are kind of in yellow are important things and tasks that I have to complete. So the first one I need to do is add a target lesion. So I've added a lesion here. I need to select what this is. Is it a primary mass, a metastasis, or a lymph node? Well, this is a metastasis. It turns out this patient has metastatic renal cell carcinoma. And I need to say, where is it located? We actually use a drop down for that because that becomes important. I don't want people to say, one person to say lung and another person to say pulmonary or something like that, or kidney versus renal. So we're going to say this is in the lung. And in this particular application, we have it active to assess for mass criteria. So we need to answer the question, does this have marked central necrosis? Now, I know it does have some necrosis here. Okay, we do see that error, but technically it doesn't meet criteria for marked central necrosis, which is more than 50% of the lesion. So I go ahead and select that. And now my cursor changed. It's ready for me to select a region of interest. So I'm going to go ahead and draw around the edge of this tumor. We'll see how steady my hand is here today. And I think that looks pretty good, and it's going ahead and selecting that. It's showing the portion of that that meets criteria for vascular tumor burden. And I'm going to select it, the lesion over here as well. And it's going to change the field of view. You're going to see it snap back to the normal full field of view. And that's a workflow issue. Our radiologists want it to snap back. Once they've made that measurement, they're ready to go on to the next tumor, and that's what they're going to do. Okay, so let's come on up here, and let's pick this as a lymph node. I'm gonna pick this as a lymph node, and we're gonna intentionally draw it wrong here in just a moment, and I'll show you why. Okay, let's pretend this was too small of a lymph node. We're gonna go ahead and select it. We're gonna say, add this lesion. This is a lymph node, so we'll select that. And the menu changes a bit for lymph nodes. This is a mediastinal lymph node. Okay, and it does not have marked central necrosis. Now I'm gonna draw it smaller than it actually is. Okay, we're just gonna draw it itty bitty like this. And it's giving me an error here and saying, hey, your short axis of your lymph node must be 1.5 centimeter. You need to find another lymph node or redraw that thing. So in this case, we're just going to go ahead and redraw it. I'm just going to go ahead and redraw it. It caught my error. It says, no, you cannot choose a lymph node that's that small. So here we go. I've redrawn it. And I'm going to go ahead and draw it over here. We're going to kind of pick up the pace a little bit here so I can get several target lesions and get to the end. So we've got that lymph node drawn. That was just one example. It was telling me that lymph node was too small and not suitable for use for RECIST and for choosing as a target lesion. So let's come on down to this lesion here that's in the retroperitoneum, kind of like invading the liver. I want to be on the right phase. You'll notice it's really washed out quite a bit in terms of the amount of contrast that's in there. Here's some clips here. We'll go ahead and zoom in. Very easy to use. I really like how this works. It's actually a little bit better than my actual real packs that I use. So this is another metastasis. We're going to say this one. We're going to say this one's in the retroperitoneum. And we'll say no. Well, we'll say no to marked central necrosis. It does have some necrosis, but we're going to say no to that. I'm going to go ahead and accidentally grab that clip. It's not a big deal. The software knows that's a clip. I'll show you that in just a moment here when I release. So it kind of says, hey, that was a clip. That's too dense. We're not going to include that. And so it knows to do that, especially when it comes to vascular tumor burden. So we designed it that way. I'm going to go ahead and select the lesion over here. It's going to snap back into shape. Okay, I'm going to go ahead and link these. It has other features that I wasn't showing you. You can certainly link images and so forth. That's not a very hard thing to do. Here's a big peritoneal metastasis. I think we'll go ahead and select that one. And I'll line these up here. They're a little bit misaligned. We'll zoom in on it. I do like the zooming feature. It does help with very accurate tumor measurements. I can tell you that it's very tempting not to zoom on when you're doing a real case because you want to move quickly as a radiologist, but this software just makes it so easy because once you do the zooming and get accurate measurements, it's going to snap back to place for you. That's part of the reason we don't like to zoom, because you have to zoom in and then zoom back out on normal PACS workstations. So this handles that very nicely. So we have a number of target lesions selected, and then there's some non-target lesions. So we need to comment on those. What's going on with the non-target lesion? It lets me answer that here, okay? There are some, so none is not correct. They didn't have complete response. They had non-complete response, non-progressive disease. So that was actually what was defaulted, but that's what we're selecting. And then let's pretend I went through all this and there are no new metastases. I can confirm that. Now notice this panel is all clear. We've finished all of our tasks. We're at about 5 minutes, Tim. Not doing too bad here. And then we're going to go ahead and hit export here. And at this point, it's going to go ahead and generate our summary report. Now, this is a new database, so it took actually a little bit longer than it normally does. But this is what it looks like. This is that summary report. Now, it's a little bit different in the look and feel of what the one that I showed you. All the same information's here. We have our target lesions. We have our coded ID. It's a fake ID for this particular patient. All of our length measurements and other metrics are here. The percent change is here. The response is here. And in this particular display, we chose to look at the raw CT images at CT0 as well as the vascular tumor burden separately. Or as I showed you in the PowerPoint, you could have merged these together and just looked at one. Or you could look at this graphically. There's a number of different ways to display this. It tells us if there's new metastases and so forth, has the data, the CT scans on here. This is just the display, though, okay? You need to remember that. I'm going to go ahead and click out of this, and it's already asking me, hey, are you ready to do the next case? Yes, I am, but what if we had an error or a query in that? Well, we could come back and reload that case. It's not impossible to do that. If I can find here, one moment here. All right. It's very possible to go back in and reload cases. And when you do that, you can fairly quickly double-click and go straight to the lesion. So for a moment, just imagine that this is longitudinal. Maybe this is a new scan and this is the old scan. It'd be very easy to find that old target lesion. You know, in real radiology practice, you have to manually scroll through to find things like that. You don't have to do that here. You're already going to easily get to these target lesions just by double-clicking on them, and then it make it very easy to measure your new target lesions. So let's call it quits on this part of the demo, but I wanted to show you where is all this information going. Now, this is the database, okay? This just has, we just did the one patient in here, and it's got a lot of information in here on the patient, their medical record number and so forth, all the length measurements. Just to give you an idea, I'm going to kind of scroll here. I'm not sure how this will show up on your screen, but it goes on and on and on and on. Tons of metrics that we're keeping track of. All this is auditable, and if we, or you're able to audit, if I can spit it out correctly. If we make new measurements on this patient for some reason, it's going to get added in as a new line. You're going to be able to track every movement that you make with this software, and that's critical in these clinical trials. So we have that, a very large, rich database. We also have the target lesions saved. Okay, I'm not going to go into them individually, but we have DICOM images of each of those target lesions, and we have very easy to access thumbnails so that, you know, it's very easy for someone to look at and say, what was that radiologist seeing? Well, you can just come here and directly look at these images and pick up exactly what someone was seeing, okay? This was the exact lesion that I measured. You could confirm that. All the metrics are listed on here. The colors are changeable. Maybe this blue's not showing up very well on your screen, but you can change those colors very easily. So that's kind of an overview of the actual software in real-life practice, what it looks like and how it functions. And so hopefully that's helpful to you to understand what we can do with this software. So I'll toss it back to you guys, Tim, for setting us up here for the next step.
Sure, absolutely. Well, thanks for doing the demo. That's great. And we are certainly getting a handful of questions coming in. So just as a reminder, while we're sitting here, we've got a few more slides to get through, primarily around validation, right? Because one of the things that, as any of you are touching the clinical trials, have talked to the folks at the FDA, they always want to know, hey, how has this been used? Well, how have you done your validation on your approach? And so we're going to talk about that now. But you know, if you have any questions, feel free to type them in the question box, and we'll get those answered for you.
All right, thanks, Tim. So yeah, we are really trying to validate this in a heavy manner. So we've already tested in a phase 2 study of metastatic melanoma, and then we also went on to, thanks to our partners from Pfizer, tested in a prospective, looked at the data from a prospective phase 3 study of metastatic renal cell carcinoma, sunitinib versus interferon. This was that pivotal trial by Motzer and group that made sunitinib the first-line agent for metastatic renal cell carcinoma. And so we were able to do a secondary or retrospective analysis of that with our software and really look at a number of different tumor metrics. And that's where our vascular tumor burden really shines, very predictive of outcome. I would argue that's currently the best biomarker in existence for predicting response to anti-angiogenic therapy. But I'll let you read that when our manuscript gets published in Radiology, and you can make that decision for yourself. So we started there with the validation. Now we have some ongoing studies that we're doing. Okay, first one is we have 12 radiologists from 11 different institutions, and they're comparing eMass to a standard of care PACS viewer. iSight is the one in particular that we're using. And what radiologists are doing is they're using manual data entry, just like you would normally for most historical clinical trials and for clinical practice, versus eMass. So they're looking at the same 20 patients, and they're going to use different reading sessions and all randomized to keep, so we don't have recall bias and so forth. And they're evaluating images per RECIST, CHOI criteria, and MASS criteria. I can't compare a vascular tumor burden between standard of care versus eMass because no one else can measure vascular tumor burden except for eMass. So they're just going to use these 3 criteria. And you can imagine you could add lots more to it, but it would get quite burdensome on my crew for the manual data entry side of it. So that's what they're doing in the trial. These are really expert readers from different universities. And it turns out, this is really important, that eMass was 50% faster than the manual data entry. Okay, so we actually got tons more information from eMass, but it was actually faster, twice as fast, 7 minutes versus 14 minutes. And that was really a nice surprise for us. How does that translate to you in the clinical trial? Well, that helps us keep our costs down. So if you're working with us as partners, that's one way we can keep costs down. We're gathering tons more information for you and doing it faster, and we can pass that cost savings on to you. So it turns out eMass also had significantly less patient-level errors. This is really surprising. Okay, not that it was better, but how actually poor manual data entry was. Okay, so there was 0% errors. The errors I'm talking about are too many target lesions, more than 2 in one organ, measurement errors, measurement data transfer errors, calculation errors, categorization errors. You would be surprised, even after training these experts, that those errors pile up when you do this manually. They're really good at what they do, and on a patient level, 30% of patients had one or more significant errors. And I say significant because we actually cut out super tiny minor errors, like if they rounded wrongly to the wrong 1/10 of a centimeter, we didn't count that one. But these are fairly significant errors. 30% of patients had it when we did manual data entry, 0% errors. Now the trial's ongoing, but we're far over half now, and there's still 0% errors with eMass. We're very pleased with that. Both eMass and iSight, are these comparable? iSight's your standard of care. Can you measure length the same using this, method that you're talking about, drawing the region of interest? Yes, we can. Actually, technically slightly better, but not statistically significantly better. Yes, we can measure length very accurately. And you mentioned this vascular tumor burden thing. Maybe you're interested in that particular metric. Turns out we're really good at measuring that. The intra-class correlation coefficient there was 0.96, which is really high, close to perfect. That probably has to do with how many data points you're gathering, a lot more pixels. It's easier to trace around a tumor than it is to measure a small tumor in terms of just a linear length. There's less variability with our vascular tumor burden than there is with simple length. So yes, we're doing a lot of validation. Maybe that's not enough for you. It's certainly not enough for me. So what are we doing here? Well, that same group of a dozen radiologists, this expert team from the Society of Abdominal Radiology that I work with, we're going to validate eMass in all of these trials listed here. We have a NIH grant that we've submitted, and that's our plan, is to go through all of these clinical trials here. Most of them are phase 3, and a couple of them are phase 2. And really, to be honest, this is not an exhaustive list anymore. We've already added a couple other trials that we're going to consider, not as part of this particular grant, but we're looking into the immune-related response criteria with one of the big pharmaceutical companies we're going to start talking to and see if we can convince them to help us reanalyze their trials for that as well. So lots and lots of validation coming with this. We're comparing the standard of care. We're testing it in a whole bunch of different new trials. I'm really excited about this tool, and that's what we're happy to have this webinar today to share that with you. So there's some future capabilities of eMass that it doesn't do precisely right at this moment, but it certainly can do. And so that's what I wanted Amit to kind of chime in on and tell us a little bit about where he thinks this could go in the future here.
Yes, so you've probably CT and MRI are both volumetric-based acquisition modalities. So the logical step for all of these criteria is to actually move to volumetric analysis. You have tumors that are actually masses that have 3D structure, and a 2D-based criteria is not necessarily going to be representative of the actual nodules or the tumors. So we've decided to actually incorporate those volumetric tools within eMass for the next release. And this is sort of going along with some of the initiatives that are out there, particularly for KIVA. So it's the Quantitative Imaging Biomarker Alliance, and they're trying to validate the use of volumetric tools for measuring these nodules over time. You also saw Andrew making these measurements with 2 different time points for the same patient, and he was zooming in and sort of matching the slices. Well, because you have these 3D volumes, you can actually co-register them within, and they're going to be slightly different depending on the breath hold, but you can put them together and co-register them to the point where if you drew a region of interest around one time point, it would actually automatically show up on the next time point. And so you can then adjust that one on that time point. So it makes the workflow much, much faster. You also saw Andrew making those regions of interest and they automatically doing the longest axis. We feel like that increases the amount of, reduces the amount of variability so that you're not having these line measurements be off by sometimes millimeters because the pixel resolution isn't that great within some of these slice planes. But as he was doing that, it automatically calculated some of the other biomarkers like the vascular tumor burden. So let's say you have a new one that you want to assess in your clinical trial. eMass is pretty flexible in the way that it actually makes those measurements, and it doesn't add to the actual workflow for the radiologist. It makes those measurements as they're making their regions of interest. So there's no added workflow for them. And then some of the other criteria require additional modalities like PET, for example. So eMass in the next release will have the ability to import in PET scans, and then you can co-register them automatically to your target modality.
Absolutely. Yeah, and everybody online, thanks for hanging with us. I'm always amazed at how many people stay to the end of these webinars. It's spectacular. So thanks for hanging in there. We've just got a couple more slides, and then we'll get to your questions. So that's super cool. The other part of this is compliance, right? And the big audit stamp comes out, and you're like, oh man, here we go, right? And
And then we talked about this in the previous few slides. There's protocol adherence that this actually enables, right? If you've got a bunch of criteria that are pretty unique, or and the radiologist has a bunch of these trials going on, it actually makes you follow the protocol in terms of the assessment. It does error correction and detection as you go along. Again, it's reducing the fact that the amount of adjudication that you would have in the trial, or the amount of queries you'd have downstream when the trial is complete. So it does auditing, again, on the fly. It does even the visual-based auditing. You get that structured report at the very end, so you can actually see, in a given case, really quickly where two readers might disagree, or what a given reader is doing for its target lesion study.
Well, I'll say something again that Amit and I gave a talk last week at Imaging and Clinical Trials Conference in Philadelphia, and I'll say what I said there because I think it got some people's attention, right? If you are using a radiology diagnostic PACS system for doing your image analysis in clinical trials, you are not in compliance with 21 Part 11. I've been in that space. I have yet to see a system that is technically in compliance with 21 Part 11. When you look at something like eMass, it aids in your compliance. And, you know, that's just one other thing you don't have to deal with, right? Because, I mean, you got enough challenges trying to get through all the other hoops. Don't worry about the compliance you have in imaging. And by the way, in case anybody was wondering, during our rehearsal, if you look at that thing in the upper right of the slide, right, it's supposed to be the audit stamp, although if you look at it just the right way, it does look like somebody's pouring chocolate, like melted chocolate in a dish, which really, it's almost lunchtime here, so that now looks more like
chocolate to me. I'm ready to move on. I'm getting hungry here.
Yeah, that's what I'm thinking. I think we're in pretty good shape that way. That's a good point. Really, there's one last slide here before we say thanks, and or maybe a couple, but look, and I don't want to beat this one to death, but why don't we just walk through it real quickly, right? You know, the potential that eMass brings now in terms of cost. You know, Andrew, you spoke about, you know, you saw things taking half as much time, 14 minutes down to 7 minutes. Relative to that, you know, what kind of training was involved there? I mean, were people like, did you have to go through a ton of training for people to get down to 7 minutes to use the system?
That's a great point. In that study, I mean, we're talking about 12 different readers from 11 different sites. Some of these people I really don't personally know until we started this study. So these aren't like people that we practice the same. You know, we all have our different practice. So we basically made a 30-minute webinar and went through just, this is how you use eMass, and that was enough. That's all it took, was 30 minutes of just, you know, structured, just I'll read this webinar, and that was everything from how to open the images to how to, you know, keep the data for the trial. So some of that was wasted space just for the trial itself, not just the software, but 30 minutes is all it took.
Yeah, and the great added advantage of eMass is that you could put in test cases that you can do periodically during the trial. So if you had a bunch of readers on a trial, you put these test cases in, you get that visual audit trail for what these readers are doing. And if someone's out of sync with the rest of the readers, you can retrain them.
Right. Yeah, that's a good point. Well, and because you've got the efficiency and that kind of stuff, it doesn't cost you a bunch of time to have somebody take a look at, you know, test cases and stuff like that. And look, the bottom line is, and you all know how this works if you've done any kind of imaging in clinical trials, you get to the end of your trial, you go, okay, look, let's gather up all the imaging time points and all the imaging data, get it to the biostats team with everything else, and then you start discovering that you're missing things. And that is the worst time in the universe to figure that out, right? And so, you know, the other thing that eMass does, and I love it, and I know it sounds silly, but, you know, my favorite part, Andrew, of your demo was watching all the yellow boxes, right? Because as the yellow boxes disappeared, it's like, okay, we're getting close to the end, but we're not there yet. And when all the yellow boxes are gone, it feels like the software is clean, and you know that you've been complete, right? And that's the important part of that.
Right, and we talked about it. We really need some cool sounds added in there, like a video game as those disappear, you know, like Bejeweled or something like that would be really cool.
That would be awesome. Yeah, that's great. How to bother your neighbor while you're reading for clinical trials in the reading room. That would be great. There'd be a whole psychological study that comes out of that. Hey, we are at the end of our time. We've got 10 more minutes, and we've got a handful of questions or more that we want to get through. So we're going to try to get through several of them. Some of Andrew are for you. Some of it might be more in your camp. But look, just as a review, we did get through that wonderful history of tumor response assessment. We talked about some of the challenges. You saw eMass. You saw the fact that what I think is really cool about it is, you know, you do these measurements, you run through that workflow, and it gives you all of the historical assessment data as well as all the new ones with new biomarkers and new capabilities, and that is actually pretty cool. So we hope you all had a good time at the webinar. Again, I'm always amazed at how many people stay to the end. It blows my mind. So let's go through a few questions for these gentlemen, and I'm going to work through the list here real quick, and a few of these are kind of straightforward. Oh, yeah, one question that's important. Will we send a link to the recorded video after the webinar? Yes. The link will be on Imaging Analytics University if you want to go find it, but we will also send out a link to the recording, so certainly don't worry about that. Let's see, we've got a couple of...
Tim, I think you said that takes a couple days before that's available too.
Yeah, that's right. Yeah, what we'll do is we'll take the recording, get it converted. We actually host it on YouTube, and then it goes through. There's like all sorts of technical things. So yeah, it'll probably certainly by the end of the week, right? At a minimum, maybe by the end of the day if we're lucky. So, but I would guess by the end of the week. Here's a good one. I think it's are the vascular tumor burden ROIs based on the Hounsfield unit thresholding?
Yes, that's a great question. It is, and it's kind of confusing because, you know, if you're measuring attenuation, you know, like for Choi criteria, in one of our studies, we found that to be enormously unreliable. Interclass correlation coefficients of like 0.21, which is rather dismal. And so you'd say, well, how could you be so high in terms of your agreement with this? So what it does is you're actually measuring the amount of tumor within a threshold range. Okay, so that's actually very reliable. So if a tumor enhances 100 Hounsfield units versus 150, it's still the same amount of enhancing tumor. And so that's why I think it's enormously reliable. Even if you change the contrast dose, the injection rate, change your KVP, all these technical parameters with CT that do affect Hounsfield units, you're still going to get the same amount of enhancing tumor. And we're testing that now in phantoms. That's what our initial results are showing, is that yeah, you can change the intensity of the image, but you still get the same amount of tumor that's vast. So it's very reproducible, but yes, it is a threshold. And that's really helpful too. That allows us to get rid of the clip. So maybe the viewer saw, hey, you didn't get the clip there. That has to do with thresholding. We said, hey, that's too dense. That can't possibly be tumor. And we do the same thing on the low end. We don't include fat. We don't include air. We don't actually include water, which is part of why you're seeing all this necrotic portion not showing up in red on the image that's right in front of you right now. So that's a great question.
Great. Here's another one from Chris as well. You know, it says your tumor metrics right now are 2D, right? They're area metrics. Is there a good reason to use 3D? It might save you from doing ROIs over the wrong slices. You know, what's the thought on 2D versus 3D, I guess? Right?
Yeah, absolutely. I think 3D is probably the way to go. It's just not been an accepted form of analysis for the FDA. And so and if you're using software to do 3D, you don't want to have to draw splines around every single slice to characterize a tumor. So there is some software.
That would be opposite of the time thing. The 14 minutes would go up to 140.
Correct. Right. Just for 1 week. So we are in the process, along with a number of other companies, to develop software that automatically detects these tumors in 3D. And that gives you more, so we've done a few studies where your longest axis on a 3D tumor can be off by 300% or more compared to the long axis made in a 2D measurement on an orthogonal plane.
Great. All right, here's another one real quick. How would a pharma company deploy this to sites for a trial and provide the training needed? Does the software speak to existing ECRS systems or does it stand alone? You want to do that? You want me to do that?
Okay, go ahead.
Yeah. So the answer is I think you've got several options, right? So one option is to take it as a standalone, as what you saw here today, integrate it into your existing ECRS system and deploy it. That's useful. That's interesting. It's a little challenging depending on how many sites you have, right? So Andrew, like for your studies, you know, you've got a small number of sites and it's an intimate research relationship. So that workstation works relatively well. Other option you've got is to deploy it through the cloud-based solution, the EDCIQ platform that we have, where you basically have that entire workstation available in a web browser, is what it basically comes down to. And that software will still transmit the data to existing ECRS, or it can be used that way. And it has all the integrated training that you need. You've got recorded web training, we can do live training. There's all sorts of training that can go around it for compliance and that kind of stuff. But so you've got a number of different ways. The other way you could do it, very honestly, is to not deploy it to your sites at all, but basically, you know, have the trial run through like an ImageIQ, and then Andrew's team uses their eMass software for the reading, right? And then you never touch the software, but you have all the benefits of it.
Right. I think that whole centralized approach is probably the ideal for almost all clinical trials. I mean, perhaps not a phase 1, but I suppose if you have a single site, you can potentially get away with using the application locally. But as soon as you go multi-site, you really have to have that central mechanism for gathering the images. You really want one source for the data. You don't want to have multiple databases all over the place. So I really think that that EDC model is a way to go for most clinical trials.
Excellent. Andrew, we got two more questions. We probably have time for 2 or 3 more questions, and then we're done, and we'll finish up and get you all out of here at the top of the hour. Andrew, the question is, can you do volume-like measurements on CNS lesions for primary brain tumors?
We haven't adapted that. Most CNS lesions are going to be using MRI, most likely, when they're asking that, and this has not been adapted to that. It's absolutely amenable to that, and that's actually one of the next steps that was listed on Amit's slide. We're talking about multimodality. That's where we're headed. So it certainly could do that. Again, it's not volumetric yet either, but that's also a next step. So we couldn't do it today, I guess is the answer. But if someone wanted to do that and wanted to head in that direction, yes, we can build that. That's in our upcoming plans to do that for sure, because I think this would be applicable to that.
Yep. Excellent, excellent. Okay, here's another interesting one. I'm not exactly sure where this one's going, but can you do near real-time analysis is the question. How does that work?
Yeah, I like that question. I think if I understand it well too, that I think what you're asking there is like, you know, you're running a clinical trial and you need an answer. Maybe the therapy depends upon that answer. So how would you get that? Well, you could have someone locally do the reads or something like that, but you can also do what you guys have set up in ImageIQ, is like an EDC, capture the images and have someone centrally provide that read. And that happens quickly. The software is amenable to that. So yes, I would say you certainly can do real time. I mean, not necessarily on the scanner, but as soon as it comes off the scanner and it's available digitally, the read can be done. So I think this can be done real time. Most people, when they say real time, we're talking about 48 hours or 72 hours or something, but it could be done faster. It's all a matter of, you know, what the need of the trial is. So I can see where that would be important for a number of different tumors, you know, and situations in clinical trials, and I think we could do that. It's amenable.
Since everything is embedded in the software and outputs as you're doing the measurements, you're not really waiting for anybody to re-enter that data into another EDC, right? It's happening live as you're making those measurements.
The other interesting part about that is, given that, right, then the potential for this is a Tim question. This will be the last one, and then we'll wrap up. So, but you know, the potential for doing retrospective analysis. You know, you've done a study, you did it, you had it read with RECIST 1.1. And that's what you reported to the agency. But now you want to make a stronger statement in your marketing statements or something like that. The ability to do retrospective analysis on images already acquired then would be relatively straightforward, because you could just put them in this thing and have a team run through it, and you get all that data relatively quickly.
You know, that reminds me, Tim, that brings up a good point. I forgot to mention this as I was saying it earlier, is that this tool is actually applicable to contrast enhance and non-contrast enhance. It actually works with that. We define the thresholds differently, but the same concept works. And we've even had cases where the first scan is contrast and the second non-contrast, or vice versa. And it does quite well in those situations. So it is a broadly applicable tool. I think back on the question on, like, can this be done real time too? I mean, think about what you're doing real time when you don't have software like this. Someone's having to quality check all that stuff. So even after your radiologist does it, you can't, that radiologist is not calling up on the phone and telling someone centrally, this is progressive disease or not, or whatever the answer may be. Someone has to go through and quality check that, make sure nothing was forgotten, all the data points are correct, decimal points are where they're supposed to be. You don't have to do that with eMass. It does that for you. So it's much more amenable to real time than probably anything else in existence at the moment.
Outstanding. Well, hey, it's the top of the hour. We want to respect everyone's time and let you all go. Thank you all for joining us. Andrews, thank you very much for joining us on the webinar. Amit, you too. I know you guys are both super busy and handling your day-to-day stuff, but it's great to share this with the community. And everyone here, if you have any other questions, feel free to contact us. We'll get the recording out to everybody, and we'll go from there. So enjoy your week.
All right, enjoyed it. Thank you so much.
Thanks.